Hello, good evening to everybody. Okay. So we'll wait for 5 minutes and we'll see if any more people will join. Then we'll start the session. Okay. Okay. So very good evening to everybody. So I'll be a PA for this particular course of human physiology. Okay. So first, let me introduce myself. My name is Tanishka. Okay. and I'll be assisting you for this course, and we'll be doing the problem solving sessions for this particular course every Friday. Okay, so let me share my screen. I hope I'm audible to everybody. Okay, so start. Okay. Okay, so as I've mentioned, so today is the first live session of this course, Human Physiology. The course instructor is Professor Nishikant Subedar from IISER Pune, and I am Tanishka. I am a PhD scholar from University of Hyderabad. So before starting the session, let us introduce each other. Okay, so okay, any one of you can start the introduction and tell your background and in which stream you are studying, and whether it's a undergrad or postgrad. Any one of you can start the introduction. You can see many people. Somebody like Varsha, Ridha, anybody can start. Unmute yourself and give your introduction. Mm-hmm. Okay. 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 Next. Hello. Okay. From where? From Bangalore only. Okay. Okay. Next. Hmm. In animal biology? Okay, okay. Okay, next. Mm-hmm. Okay, okay. Next. We have so many people in the call. Next. Okay. Okay, so I can see there's a diversity in the class, many different streams. Okay. Anyone from zoology background or animal biology background? Yes, no? Okay, so let's start the session for today. So in this particular session, yeah. Okay. So we will be studying about homeostasis today, and also we'll be talking about the muscular contractions. Okay. So anybody wants to tell me what is homeostasis? Anyone? Yes. Yes. So homeostasis is a property where the body maintains a stable environment inside, and despite of there are certain changes in the external environment. Okay. So let me, okay. So if we'll start with this. So there is a set point. Set point senses the disbalance or imbalance um, in a stable condition on our body, and this set points give a signal which senses that imbalance, and that will give a next signal to the control center. Okay, and that control center will release certain chemicals that will work on the effector, and then that variable is regulated, and the sensor is again senses the balance regulation. Okay, so if we'll see here, so the all homeostatic control mechanism have at least three independent components for variable being regulated. So a receptor, control center, and effector. So receptor is the sensing component that monitors and responds to changes in the environment, either external or internal, and the receptor include thermoreceptor, mechanoreceptors, there are baroreceptors. So the type of changes that is happening, that is being sensed by a certain receptor, and then it gives the signal to the control center. So that is includes the respiratory center, renin angiotensin center. So the basic uh, body that controls over that particular response. So there the signal is being sent. And then there is an effector organ, or the effector is the target acted on to bring the change back to the normal state. Okay. So here is the chemical composition of extracellular and intracellular fluids. Okay, so say here we have a cell, and inside the cell, whatever the cytoplasmic material and the organelles and everything, that is an intracellular fluid that is present in the intracellular fluid. Okay, and then we have extracellular fluid. Okay, so this extracellular fluid includes the so every organ is connected well with the blood vessels. Okay, so in the blood vessels we have, along with the RBCs, we have the plasma. So that is a fluid, okay, that is called intravascular fluid. And then between the cells and between those blood vessels, the intermediate space, there also some fluid is present, that is called interstitial fluid. Okay, so there is a difference in the concentrations of this intracellular and extracellular fluid, and those are reflected in this particular uh, table. Just give me a minute. Write this down. Just a minute. Okay. Okay. So for now, if we'll see here, this is extracellular fluid. Here you can see, if you'll see the sodium concentration in the extracellular fluid is 142, and here in intracellular fluid it is 10. Somebody join, it seems. Okay. So here in sodium it is 142 milliequivalent per liter, and inside the intracellular fluid it is 10, and it is 10 milliequivalent per liter. Similarly, you can see there is a huge difference between the potassium concentration also in the extracellular fluid and the intracellular fluid. Outside it is 4, inside it is 140 only. Similarly, here also you can see in case of glucose, the outer in the bloodstream or the extracellular fluid, the glucose concentration is remained at 90 mg per 100 ml of the blood, and inside the cell we have 0 to 20 mg per 100 ml of the blood. Okay, so there is a huge differences, and the cells should maintain a certain limit of the particular components. However, the extracellular fluids have a very different uh, composition of the extracellular fluid. However, overall, when you calculate the osmolarity, there is a balance in the osmolarity between the extracellular and intracellular fluid. Okay, so for example, okay, so for example, let's start with the glucose concentration here. So as you can see, 0 to 20 mg per 100 ml of the blood. So the minimum concentration of the glucose should be maintained of 20 mg per deciliter in the blood. Okay, if that glucose concentration goes below 20 mg, that is a problematic situation, and that can be sensed by the sensor organs. Similarly, outside of the or in the fluid or in the extracellular fluid, the glucose concentration is maintained at 19 mg per 100 ml of the blood. So 19 mg is a good, 100 mg is also good, but if the blood glucose level increases to more than 110, more than 100 or 110 or 120, that is also a very dangerous situation. So there should be a balance. The glucose concentration range should be at least 20 mg per deciliter in the intracellular fluid, and it should be at least 90 mg per liter, not more or less, in the extracellular fluid. And these kind of balancing can be done here. So as I have mentioned, the osmolarity, you can see in the extracellular fluid and the intracellular fluid, the osmolarity remains same. Okay, so let's understand the homeostasis with the example of this um, blood glucose level. So the normal blood glucose level in the bloodstream, as I have mentioned, it is 90 mg glucose per 100 ml of the blood. Okay, so if the blood glucose level raise after eating, so when you take the meal, you take the glucose, do the glucose intake, and upon the glucose intake, the glucose level increases in the blood. So from 90, it increases 100, 110. So that is being sensed by the body, and there the organ which comes in the picture is the pancreas. Okay, so the pancreas have certain special kinds of cells. What are those cells called as? Can anybody name? In the pancreas, yeah, tell me. Islets of Langerhans. Yeah, islets of Langerhans. And those have how? What are the different type of cells? Alpha cells, beta cells. Yeah. So we have alpha cells, we have beta cells, and what is the function of these cells? Alpha cells produce glucagon, beta cells produce insulin. Yes. So alpha cells, they produce the glucagon, and beta cells, they produce the insulin. Okay. So when the blood glucose level increases, pancreas releases the insulin from the beta cells. Okay. And this insulin has a receptor on the different cells of the body. Okay. So when in the bloodstream, if I'm writing glucose as G, so the concentration is increasing in the bloodstream. The signal goes to the pancreas, and the pancreas, we have these beta cells which releases insulin. Okay, so this insulin is having receptor on the different cells of the body. For example, here in the liver cells and in muscle cells. Okay, so the insulin has a receptor over those cells, the insulin receptor. So this receptor, when the insulin is sensed by the liver cells or other cells, it opens the channels present on the cell so that glucose can enter, so that high glucose which is present in the bloodstream, so that glucose entry channel will open and the glucose comes in the those particular cells. Okay, so by the insulin the signal is given. Okay, and so the liver takes up the glucose and stores them in the form of glycogen. Similarly, the other cells also take more the glucose. So overall the increased concentration of the glucose is now balanced or brought it back to the normal level, which is the homeostatic level, which is 19 mg per glucose per 100 ml of the blood. Okay, then if there is a drop in the glucose level, okay, if there is a drop in the blood glucose level below a set point, okay, so then the pancreas releases the glucagon from the beta cell. Okay, so then we have this uh, in the from the glucagon receptor, and with those glucagon receptor the enzymes that is involved in the breaking down from glycogen to glucose that gets activated. Okay, and then again those glucose is released in the bloodstream, and then again the blood glucose level is brought back to the homeostatic level. Okay, so altogether the liver and pancreas regulate the concentration of glucose in the extracellular fluid. So this is one of the example of the homeostasis for maintaining the blood glucose level. Okay, so homeostatic mechanism of the major functional systems. So here the essentially all organs and tissues of the body perform function that help maintain these relatively constant conditions. Okay, including gut, including kidneys and other arteries, lungs. Okay, so for instance, lung provide oxygen to the extracellular fluid to replenish the oxygen used by the cells. Okay, so lung provides the oxygen. Similarly, the gastrointestinal system provides nutrients. The kidney regulate concentration of the hydrogen, sodium, potassium, phosphate, and other ions in the extracellular fluid, and extra urea is being excreted from the kidneys. Okay. So, in regulating other control system or homeostatic mechanism, we can also understand by the temperature maintenance of the body. Okay, so here the brain is the control center. Okay, so what happens when there is a condition? Okay, so when there is a condition, suppose your body is feeling too cold. Okay, if you feel cold and the outside environment of your body is cold, okay, then uh, inside our blood vessels, what happens? The control signal, okay, so the control center gives the signal that the cold signal to the blood vessels, so to the skin. So here the receptors are present and the skin senses this signal given by the brain of being cold, and in that condition what happens, the blood vessel, so that controls the blood vessel constrictions and dilation. So heat is conserved by blood vessel constriction and to inhibit, and after inhibiting the sweating. Okay, so once the signal is received of feeling cold, then the blood vessels get constricted. Okay, they get constricted and also prevent sweating. So when the blood vessel get constricted, then the temperature in the blood increases. Okay, the temperature increases, and then again the signal reaches to the brain, that and that's how the entire equilibrium is achieved. Similarly, here when there is a feeling of being hot, the brain senses that feeling, okay, and that hot feeling and is reported to the skin, and in this particular condition the blood vessels dilate and induce the sweating, and with the sweating the blood temperature again decreases, and from hot it uh, cools down and come to the normal temperature. So that 37-degree temperature, body temperature, is being maintained by the homeostatic mechanism. So in regulating body temperature, there are temperature receptor in the skin which communicate information to the brain, which is control center, and the effector is our blood vessels and the sweat glands in our skin. So that is how the control of body temperature takes place. Hmm. So this is the again the same thing, the homeostatic temperature, which is 35.6 degree to 37.8 degree. That is the normal body temperature. So if there is an imbalance, okay, the body temperature increases when exercise or in the hot climate, the blood gets warmer in the hypothalamic set point. Okay, so the temperature, so when the change in the temperature takes place, it is sensed by the signal goes to the brain, and that is also in the hypothalamus because that is a sensor for the temperature. So the so this particular region activates the heat loss center in the hypothalamus, and hypothalamus give instruction to the skin. So skin has and the sweat glands are activated, secrete, and also here the blood vessels they dilate, and then body temperature is decreased. The opposite happens in the case of cold temperature. Okay, so this was the homeostasis part. Along with this, we have also uh, okay. So along with this, we also will talk about the muscle contractions. So we have three types of muscle. Okay, we have the cardiac muscle, we have the skeletal muscle, and we have the smooth muscles. So okay, so So we have, I was talking, we have skeletal muscles, we have smooth muscles, and we have cardiac muscles. So broadly, we can divide the muscles into like involuntary muscles and voluntary muscles. Okay, so involuntary muscles are the all our organs, that includes our organs, because that our organs are functions independently, they are not with our will. So those are the involuntary actions that is being done, or involuntary muscle contractions. But the skeletal muscles perform the action which are with our wish, and those are referred as voluntary actions, and those are done by the voluntary muscles. Okay, so uh, so this is a section, I can say. Okay, so from next time, please join the class on time. This is already 30 minutes late. Okay, so this is a you can say this is a muscle, a skeletal muscle. Okay, so this is a skeletal muscle which is we refer as muscle fasciculus, and this muscle is made of muscle fibers. Okay, so these are the muscle fibers. So individually you can see these are the muscle fibers, and then when the sectioning is done of muscle fibers, these muscle fibers are also made of myofibrils. Okay, so these are myofibrils. And in the myofibrils, if you'll see in the detailing, you will see that there are some dark zones and also light zones. So it appears in fragments of dark and light and dark and light. Okay, so it appears like that. So in the myoblast, these are the these are the myofibrils. So in these myofibrils, if we'll see the sectioning, so it will appear like this. So it appears, okay, dark, then light, then dark, then light, then dark. That is what its appearance is. Okay, so here in these particular light and dark and light areas are referred as certain zones. Okay, so the light zones are referred as eye band. The portion of the muscle which is appears light, that is referred as eye band, and the portion of the muscle or the myofibril which appears dark, which is that is referred as a band. Okay, so the reason we are being here is in the A band area, when the light is put uh, uh, like through the way of the A band, then the refractive index that it gives, that is a certain value. And when there is a on the right angle, when the light is put, and then the refractive index that is produced, that is a different. So that is a property of these dark zone, and that is referred as anisotropic. So getting two different kinds of refracting index, that is why this particular region is referred as anisotropic, and that is why it is referred as A band. However, in this I band area, there is no change in the refractive index of the lights. That is why it is referred as isotropic. So that is why it is referred as I band. So there is A band, which is the dark band, and then there is the I band, that is the light band. Okay, so in the okay, so in this um, myofilament, so we have now seen A band and I band. Then when it has been looked closely, you can find a zigzag line present in between the I band. Okay, so in between the I band, okay, so here if I'll draw, they found a zigzag line between the I band. This is the dark band, light band, and light band. Okay, and yeah, and this zigzag line is referred as Z disc. Okay, so uh, so then we have taken, or it has been observed, that if we'll take from this one Z disc to another Z disc, it covers half of the I band, one full A band, and half of the I band. Then again, then half of the I band, one full A band, and then again half of the I band. Okay, so that is what is being considered as unit of the myofibril unit, and that unit from one Z band to another Z band is referred as sarcomere. Okay, and that also later proved to be the functional unit of the myofibrils. Okay, so these are the referred as sarcomere. Here also in the picture you can see from one Z disc to another Z disc, it is referred as sarcomere, and this is the unit of myofibril. Okay, and this sarcomere contains half of the I band, one full Z band, and another half of the I band. Okay, so and also in between the A band you will have a middle line that is referred as M line. Okay, then see, next, so next when it has been observed, so the light band from this sectioning also, if I can show you, so when there is a thin film cross sectioning done at this particular position here, then it gives the section looks like this. Okay, so this is a thin filament. So these, when the close electron microscopy has been done, these are the proteins present, a fibril protein present in the thin filament, and these proteins are actin proteins, and that is why these I bands are made of the actin thin filament. Okay, then when there is a sectioning done here at this particular position in between, like near the M line, okay, there it has been observed. So this is the protein where, so in the, for now you can understand in the thick. Okay, also one more thing. Okay, so as I mentioned, this I band, which is a light band, and it is made of actin filaments. Okay, these are made of actin filaments. However, and these are also referred as thin filaments. Okay, so these actin filaments are referred as thin filaments. So these are actin thin filaments. Okay, so this is a myofilament within a sarcomere. So the sectioning done here, it shows all the actin thin filaments. Then the sectioning done here, it shows the uh, thick proteins. So those thick proteins are nothing but myosin proteins. Okay, so that is a myosin protein. So that is a myosin filament. That is also we can refer as thick filament. And then in between this area, in the dark band, when the section, so this is all the dark band or A band. When the sectioning is done in this area, there is, we can see that both the actin filament and myosin filament both are present in this particular area. Okay, so here this sectioning you can see. So this is the sectioning done at this particular portion. So here there is an overlapping of both actin and myosin filaments. So the beauty of this particular sectioning, you can see here that these small dots are the, if I can just put them red, and red, red one, you can see these are the actin or thin filaments, and the yellow bigger dot is the myosin filament. So if we'll see geometry wise, all the myosin individual filament, you can see they are surrounded by six uh, actin filaments or six thin filaments. Similarly, if you see uh, one particular this actin or thin filament, it is surrounded by three uh, myosin filament. So that is a beautiful geometry of this sarcomere, and it is always you'll find like this only. So yeah, so this is the same thing. So we have seen this is a muscle cell, and the muscle cell, this is a myofibril we are looking at, and this is a sarcomere, a unit. So it appears like a dark band, a light band, a dark band, and a light band. So there we have a defined functional unit that is referred as sarcomere. Sarcomere covers half of the I band, a full A band and again half of the I band. So this sarcomere is starts from one Z disc to another Z disc. Okay, and here you can see these are the Z disc. It appears in a zigzag form because this is a state, this is a position where from where the actin filaments uh, uh, extend in two different direction. Okay, so suppose, so this is the disc. Here you can see, so this you can see this at this point one actin filament is protruded here, and in this direction one actin filament is protruded here. Okay, so similarly here another actin filament, here also another actin filament. So that is why as it stretches towards us one side, there the it appears at one Z appearance. Okay, if it is in the right side, the actin filament that is being extended, it is in the right side, then the zigzag is in the right side. So that is how it line is made. Okay, and then so this is a thin actin filament, and then we have this thick myosin filament. Okay, so these myosin are not only one individual filament. Okay, so these myosins are uh, 200 to 400 individual myosin filaments, and these are stacked together on one each other. So these are also in a such a shape, you can see like this, the shape is like this, and several actin filaments, myosin filaments are stacked. 200 to 400 actin filaments are stacked here in these. Okay, so these are stacked like this, and these area is referred as tail. This, yeah, so this is referred as tail, and this is referred as head. Okay, this is tails, and these are referred as myosin heads. Myosin heads. Okay, so however here this one is an actin filament, the top one. So uh, so we understood myosin structure. Then we have the actin. So actin is a globular protein. So this is one actin. So this is a globular protein. So that is why it is referred as G actin. Okay. So many such G actins, okay, uh, come and bind with each other and make a filament, and that is F actin. So this particular whole stretch of actin, globular actin, is referred as actin filament. So this is referred as actin filament. Okay, so this is an actin filament, and on these individually on all the G actin we have the let's say we have the actins active sites. So on these actins we have a site for calcium binding, or you can say like these sites are present for the myosin head binding. Okay, not calcium binding, sorry, it is for myosin head binding. So actin binds to the myosin molecule with this particular sites present in the G actin. Okay, but these sites are covered by the molecules which are or the a thread which runs along the actin filament, which is tropomyosin. So tropomyosin covers those sites where the myosin molecule can bind. Okay, and along with the tropomyosin we also have a troponin protein, so that also we'll talk. Okay, so when the muscle contraction happens, so what happens, uh, so this is a sarcomere, you can see. from one Z disc to another Z disc. So the in the muscle contractions, there is a decrease in the length of the sarcomere. Okay, so that means both actin and myosin, they slide over each other and bring both the Z discs closer, and also decreases the sarcomere length. Okay, so that is how the um, muscle contraction takes place. So how exactly this, during the movement, or during the muscle contraction, the movement between the actin and myosin filaments, or the binding happens, that we'll be seeing now. So this is the first step. Okay, so this is the myosin. So in the, okay, yeah, so this is the globular actin. Here you can see this is the myosin binding site, and this is the ATP binding site present on the actin, and there is a this on the myosin also we have the actin site. So on the globular actin we have the myosin binding site, on the myosin we have the actin binding site, and also we have the ATP binding site. So when there is a calcium release, okay, when there is a calcium release, calcium interacts with the troponin, and troponin pushes or removes the tropomyosin from the G actin, and the G actin active site, or the myosin binding site, get exposed, and that's how the myosin can bind to the actin with this actin binding site. So this is a rigor state. Okay, so this is a rigor state. Okay, this particular state where the myosin is bind to the actin is referred to the rigor state, and it is a transient state usually. Okay, so then after this particular actin myosin binding, what happens? The ATP binding happens. So when the ATP binding happens, it causes the conformational change in the myosin, and then myosin detaches itself from the actin. Okay, so this entire cycle, uh, okay, we'll talk about later. So once the ATP binding happens, it dissociates myosin from the actin, and this is we also refer as cross bridge, and the cross bridge can now go through the cycle on a new G actin molecule. So it dissociate from that particular actin, and then what happened? ATP hydrolysis. When the ATP binds, ATP hydrolysis takes place. So myosin ATPase hydrolyzes the ATP to ADP and phosphoenol, an inorganic phosphate. Sorry. So energy from the reaction is transferred to the cross bridge. ADP and PI remain bound to the myosin. Okay. Then the myosin head moves to the COG position and binds to the G actin molecule. So here you can see, till now the actin, so the myosin is in a tilted position. It is an acute angle, making an acute angle. When the ATP hydrolysis takes place, then it binds to the actin in a right angle. Okay. Then what happens when, then there is a phenomenon referred as power stroke. So in the power stroke, what happened? Myosin attachment to actin triggers a rapid inorganic phosphate release. So as after the hydrolysis, the um, myosin binds back to the actin, this causes a power stroke and releases the inorganic phosphate. Okay, so actin filament is moved about 10 mm towards the center of the sarcomere, okay, by the slight movement. Okay, so it moves, again it moves 10 nanometer towards the center of the sarcomere. So and then what happens, the myosin head unbinds ADP, and the remains tightly bound to the actin, that is again called a rigor state. Okay, then again one more ATP comes, and then again the cycle starts, and that movement towards the closer toward the sarcomeres takes place, and that is how the muscle gets contracted. Each ATP causes the movement of the actin myosin towards the sarcomere to the 10 nanometer. Okay, so that is how the movement takes place. So when there is no ATP present, the actin and myosin remain bound to each other, and that is a rigor state. However, this rigor state, as I mentioned earlier also, it is a transient stage in a live human being. There is a condition in the dead human, so that is a condition called the rigor mortis. So during the death, what happens, whatever uh, binding of actin myosin has been taken place, that remains as it is because of the unavailability of the ATP. So actin and myosin remains bound to each other, and that is a rise of the rigor mortis state. That is the rigor mortis stage. Okay. And when after some time, when the body start decaying, the actin myosin they start degrading, and then the muscle again go relaxed stage. Okay. So however, in the living body, because there is a continuous supply of the ATP, there is this particular condition, which is rigor, is only a transient condition, that is a very short lived condition. Okay. And allows the muscle contraction to take place. So this is the entire cycle for the contraction of the muscle. Okay, so let's talk about how this contraction, how this muscle contraction gets a signaling. Okay, yes, so okay, so what happens when we move our muscles? So we, all these muscles are voluntary muscles, so these are done by our uh, will. Okay, so when we want to do the muscle movement, our brain sends the signal. Okay, and that signal is traverses through to our muscles via the neurons. Okay, so and the brain also, I'm talking about this, like the central nervous system of the brain, which is the CNS, where the vertebral, so these are the vertebral root. Here you can see, so in the section of the spinal cord, you can see these are the, inside this, the gray matter, the gray horn is there. And after that we have the white column. Okay, this is the dorsal root, and here this is the neuron coming out from there. So this neuron is going to the skeletal muscle. So the signal is given by the central nervous system to the or you can say the spinal cord to the this particular skeletal muscle for the movement of the cell. So here you can see the motor neuron is taking the signal uh, to the skeletal muscles, the muscle fibers. And in the muscle fiber, what happens with the signal of the neurons, the action potential rises. So the signal is carried in the form of neurotransmitter. Okay, so if a nerve is carrying a signal, it is carried in form in the neurotransmitters. Okay. Okay, so okay, so we have talked about how the muscle contraction takes place. Okay, what is the composition of the myofibril muscle fibers? So individually, we have seen the composition of the muscle fibers. We have talked about the appearance, a dark band and a light band. Then the unit of the myofibril or the myofilament, which is a sarcomere. And in the sarcomere, it starts with a Z disc and ends with a Z disc, and that is a functional unit of the muscle. Okay, and then we have the thick filament and thin filament. In thin filament, we have the myosin filaments. In thick filament we have myosin, and in thin filament we have actin filament. And that also we have discussed. This is the actin and this is the myosin. So actin are several myosin molecules joined together to make a myosin molecule, uh, which is divided into heads and tails. And then we have the actin. So actin is a uh, filamentous actin. Actin filament refers as actin, which is covered by tropomyosin uh, uh, by to its active site, which is the myosin binding site. Okay, and when there is a calcium release, then only the troponin removes the tropomyosin and makes the uh, uh, myosin binding site on the actin available for the binding of the myosin, and also the sliding movement so that the muscle can be contracted. So we have understood that also through this cycle. And then how the signal reaches, how this particular contraction starts. So for that, we the signal is sent to the skeletal muscle in form of a motor nerve. So motor nerve gives the signal in form of neurotransmitters. So here is this junction. Okay, so here's this synaptonal junction here. So these are the vesicles that carries the neurotransmitter signal, that is acetylcholine. Then with the exocytosis, the release of acetylcholine happens in this particular synaptic cleft. Okay, so in the synaptic cleft we have the receptor, acetylcholine receptors. Okay, and when, hello, anyone trying to see speak anything? Okay, it's Ravindran. Anyone want to ask anything? Okay, so yeah, before initiating this part, hmm, okay, so also one more thing. Okay, so these myofilaments, okay, these myofilaments are usually covered through the connective tissues. Okay, and also in these along with the connective tissue, like around this myofilaments or myofibrils, we have the endoplasmic reticulum covering in these kind of cells. Okay, so these myofibrils are surrounded with the endoplasmic reticulum. Okay, these in the endoplasmic reticulum, and also there is a structure present in this particular um, myofilament that is referred as a T tubule. So there is a kind of depression present here, okay, which traverses here, and that is a T tubule. Okay, so this referred as T tubule, and this particular T tubule is very important for the supply of calcium. So let's connect that T tubule story to here. So what happens, I can say, is so as I mentioned, the motor neuron gives the signal in form of acetylcholine release. Acetylcholine releases and acts on the uh, muscular cells. So on the plasma membrane of the skeletal muscle, we have the receptors for acetylcholine, and once those receptors get activated, it opens a channel, sodium potassium channels. Okay, so we have seen that in our beginning slide, the sodium concentrations present outside the cells, like extracellular fluid, the concentration of the sodium, and in the intracellular fluid, the concentration of sodium. What was it? Can anybody tell? Hmm. Okay, so here you can see in the extracellular fluid, the sodium concentration is 142, and in the intracellular fluid, it is 10. Okay, so in the outer fluid, the sodium is 14.2 times more than in the intracellular fluid, and opposite for the potassium. In case of potassium, intracellular fluid has more potassium concentration, and the uh, um, extracellular fluid has 30 to 40 times less potassium concentration. Okay, so because of this difference, when the sodium potassium channel opens, there's an exchange or movement of sodium ions from outside to inside, because in the extracellular fluid we have higher concentration of sodium ions, and in the intracellular fluid we have the lower concentration. Okay, so uh, so once the sodium enters inside through the sodium potassium gated channel. So so the inner uh, so previously the inner what you can say, the layer of the plasma membrane, the inner side was negatively charged. So as the sodium concentration increases, because sodium is entering from extracellular fluid to intracellular fluid, then the charge is also changed. So from negative, it has come it has become a positively charged from the inside. So there is a changes in the charge. So that is called as depolarization of the membrane. Okay, it's returned to uh, the opposite charge. So it is referred as depolarization state. So as the sodium enters, it makes the negative charge to a positive charge, causing the depolarization of the plasma membrane. Then, as I have, you can see here, there is a T tubule. This depression here is T tubule. Okay, so in this T tubule, we have channel. Okay, in the we have a certain channel of the membrane, and also I have mentioned there is a this particular area is endoplasmic reticulum. Okay, so this is endoplasmic reticulum of the cell, and this is the plasma membrane of the muscle cell. Okay, and here only we have these our actin and myosin molecules present. Okay, so till now we understood in terms of plasma membrane of the skeletal muscle or skeletal myofilament. Okay, so sodium enters, creates the positive charge on the particular plasma membrane of the skeletal muscle, and then we have in the deep in the T tubule we have a channel which is a uh, voltage gated calcium channel. Okay, so we have a voltage gated calcium channel. Okay, this is also so this is also referred as dihydroxypyridine channel, DHPR. Okay, because it particular channel has the affinity for this DHPR molecule. So that is why some people refer as a DHPR channel as well. Okay, so and this channel is linked to another particular channel which is referred as yeah, so this is a ryanodine channel. Okay, so this calcium voltage gated calcium channel is present on the plasma membrane, and on the endoplasmic reticular membrane we have a ryanodine channel, and these two channels are linked with each other. Okay, so in the normal condition, when there is a negative charge on the plasma membrane, these two channels linked with each other, and this ryanodine channel remains closed. Okay, but when there is a change in the action potential, there is a depolarization of the plasma membrane. Due to the positive charge of the plasma membrane, there is a change in the conformation of the calcium gated channels. Okay, and due to that uh, this ryanodine channels, it becomes open, and inside this endoplasmic reticulum we have enough calcium, and that is now released in the uh, intracellular fluid of the uh, but in this myofilament in the muscle cell. Okay, so then there is a release of the calcium, and when the calcium release, it binds to the troponin, and tropomyosin moves to expose myosin binding site on the actin. Okay, and then cross bridge go through the several cycles as long as calcium remains bound to the troponin. Okay, and then what happens as the this wave of depolarization. So as this all this channel opening happen because of the depolarization. As the depolarization ceases, the again there is a change in the conformation of the calcium channel, which again links with the this particular iodine channel, and then again the movement of calcium ion also stops. Okay, and whatever calcium was came, that also it is pumped back to the ER, because there is always a equilibrium needs to be maintained. So again there is a calcium pump present in here in the wall of ER or sarcoplasmic reticulum. So it again pumps back the calcium. Okay. So as the ATP dependent calcium pump decreases the calcium concentration, the cytoplasmic calcium leaves the troponin, and the tropomyosin blocks the myosin binding site on the actin, and the contraction ends. So that is how the entire process is regulated by our brain, and it sends the signal in form of the motor neuron and releases the neurotransmitter, and with the help of neurotransmitter the entire polarization changes in the conformation that causes the release of the calcium ion, and because of the calcium ion presence only, the actin myosin binding takes place, and due to that the sliding movements of actin myosin occurs that causes the contraction of the muscle. So this is the entire excitation and contraction coupling pathway. I hope it was clear. If you have any doubt, you can ask me now from this one and also from the previous one. Anybody wants to ask anything? Okay. So this is the summary. The first one is the action potential in the muscle membrane. The axial potential arises, which causes the depolarization of the T tubules, causes the positive charge, and it opens uh, uh, the channels which causes the sarcoplasmic reticulum calcium release channels, which is ryanodine receptors. Okay, which increases the intracellular calcium concentration. Calcium bind to troponin, tropomyosin moves and allows interaction of actin myosin, and the cross bridge cycling and force generation takes place, and calcium re accumulated by sarcoplasmic reticulum, and which causes the relaxation of the muscle. So this is the entire cycle. Okay. Okay, so this was the content of the first week. Anything else you want to ask me from whatever we have discussed through? Any doubt? Okay, if nothing, then let's see. There are a few problems. Okay, so this is one of the okay, this is one of the explanation. So you have to tell or find the correct label pair. So here, so we'll start with the third one. So third one is the homeostatic condition. Okay, so what will be the third one? This is a image referring to the blood glucose level, and it's homeostasis when the glucose level rises, when the glucose level goes down, and how, which hormone is related, what is the effector organs. Okay, so okay, so the okay, let's start with the first one. Okay, so pancreas. So what does pancreas do? It releases insulin. Yet it releases insulin. So first one will be to release of insulins by the pancreas, and then after the insulin is released, what happens? The second step is yes, because release of insulin activates the glucose channel, glucose uptake to the muscle cells and liver cells. Okay, so the glucose enters the cells. So glucose uptake is increased. So when the glucose uptake is increased, the glucose level in the blood goes down, right? Then what happens? Yeah, then what happens? Yes, and after okay, so when so okay, so this is the homeostatic condition. Then it is shown that the blood glucose drops below a set point. Okay, so the glucose level in the blood is less. In that condition, with what does pancreas do? Yes, it releases glucagon, and glucagon, what does it do? How? No. uh, Glucagon's function is to activate those enzymes in the liver cells, and that converts glycogen to glucose. Okay, because when glucose uptake also takes place in the liver cells, what they do, they store glucose in form of glycogen. Okay, so when there is a need of glucose in the bloodstream, when the signal is gone to the liver activates, or glucagon activates such enzyme which breaks down the glucose to the break down the glycogen to the glucose, and then the glucose is releases to the bloodstream, and then the blood glucose level increases, that has been dropped down and come to the homeostatic level. Okay. Okay, so there are, yeah, there also you have to do a labeling. Okay, so okay, first you tell me these globular green dots, what are those? First one. Yes. Yes. So these are globular protein. Individually, these are called as, yes, it is globular actin, G actin, and when they polymerize and make a filament, that is called as actin filament. So the unit of actin filament is G actin. Okay, and this uh, helix line protein that goes along with the actin, it is nothing but tropomyosin. Okay, so what is the function of tropomyosin? Can you speak a little uh, high, loud? uh Not actin filament. So these so in the globular actin also you can see these sites, right? So these are sites are for myosin binding, right? On the globular actin, these sites are present, and these sites are for myosin binding. Okay, so even though the myosin is there, okay, and myosin has also has this actin binding site, but the myosin cannot bind to this actin because the actin's myosin binding site is covered with tropomyosin. Okay, so the function of tropomyosin is to cover the myosin binding site on the actin. Okay, the contraction happens when the actin and myosin contacts with each other, right? When there is a sliding movement, and that happens due to their contact. But during the uh, so as it is a voluntary action, without the signal cannot, anything cannot happen on its own. So when the signal is not present, to avoid the binding of actin and myosin with each other, tropomyosin traverses through the actin filament so that the sites for myosin binding remains blocked. Okay, is it clear, or should I tell again? Okay, so okay, so tropomyosin function is to block the myosin binding site on the actin. And the third one, sorry, so then the second one, which is troponin. What is the function of troponin? Hmm. Hmm. It binds to the calcium, right? And after binding with calcium, what happens? So once the troponin binds to the calcium, it moves the tropomyosin away from the actin, so that the myosin binding site on actin gets exposed. Okay, so it unmasks the myosin binding site on the actin, okay, by removing the tropomyosin. Okay, okay, so so hmm. So this is a motor neuron connecting to the skeletal muscle. Okay, so this also you can do, you can tell me. So the first, this is a section of the spinal cord. Okay, so in this section of the spinal cord we have the gray horn. Here this is the gray matter. Okay, first one is the gray horn. Okay, next we have you can say, after that we have the second one. What is the second one? Sorry. Yes, and then third one. Yes, and then the fourth one. Yeah, that is a sensory neuron. Yeah, so that's it. And majorly we have discussed the two important things are this cross bridge cycling of actin and myosin binding and how the sliding movement takes place. And as long as the ATP supply is there, uh, the movement of this thick and thin filament, which brings these uh, the sarcomeres lines closer to each other, decreases in the size and causes the contraction of the muscle. And also this excitation and contraction coupling is also very important. So that also I explained. If you have any doubt, you can ask me now. If any part you didn't understand, there's any question, you can ask me now. Is it clear? Is this excitation contraction coupling part is clear? Okay. Okay. So if no one is having any doubt, I would conclude today's session. Okay. Okay. So see you next week then. I'm concluding the session now. Thank you.